Hello, so we have a assignment today. This should be beginning on Thursday, and uh, our goal is to finish this on Friday. You're going to be with Miss Reed and work with her as you need. Use this video and work together as a team to complete the task. A lot of this is a bit of review for Chapter 1. You know, ideas like control group, experimental group, um, independent variable, dependent variable. And it requires a little bit of reading, like we've done with our MEL activities, where you read some information and then you come up with an idea, a claim. You use what you read as evidence. And in that same regard, it's like our close reading assignments, like what we did with the marijuana and insomnia video or worksheet, where we read some information and then we answer some questions. So use the information you read to answer questions or to support your own ideas on something. And the reading and using the reading as evidence is later in it, but let's focus on what the task is initially, and that is going to this website right here by Googling Virtual Lab Population Biology, or more simply Paramecium simulation. So we have our worksheet, but it's based on this activity right here. So if you don't, when you click on the link after Googling Paramecium simulation, if you don't see a page that looks like this, you're in the wrong simulation. So let's go there right now. And as you can see, I Googled Paramecium simulation, hit enter, and bam. The first choice is what you need. So you just click right here. And here we go. This is the setup. Let's see though, what do we need to do next? So I'm going to go to the worksheet to figure out what I need to do. All right. So it says the first thing is to read and summarize the purpose, objectives, and procedure. And read and summarize basically the portion on the left side of the screen for the simulation. And that's right here, purpose, objectives, and procedure. So basically, before you start, know what you're doing, come up with a plan, understand what the simulation's about by reading these three sections here, purpose, objectives, and procedure. It says, what's the scientific question that motivates this experiment? Well, the scientific question is the scientific problem, same thing, and we always start science with some kind of problem or question. Here, it's right at the top right here. It says, how does competition affect population growth? So that's exactly what you'd copy over here. The main question that this experiment seeks to explain or find answers to is how does competition basically affect the size of populations of a species? Is it good? Is it bad? Does competition have no effect on the size of population? This experiment will show you. All right, so the next part is read the purpose. So you read this purpose right here and develop hypotheses, or I'm sorry, hypothesis that attempts to answer the scientific question above. So read through this purpose here, and it mentions that you're going to be working with two different species of protozoan. These are single-celled organisms, but the first part of their name is Paramecium, and you have two different species, so it's going to be Paramecium aurelia and Paramecium caudatum. You'll find out later. You'll get more specific with the species. But right here you put my hypothesis equals, and then right here, what do you think your answer is to the to this beginning question? So your hypothesis is, I think competition will affect population growth. You could put that here. I think competition will not affect population growth. Or you could come up with your own hypothesis. But you're seeking to explain. Remember, you're making a claim, a confident statement about how you would answer this question. And then later you can go back to the hypothesis, see if you were right or not, just based on the results. All right, so the next part is summarize or define the following terms or paragraph. So we need to define what paramecium is. So you can just Google paramecium and then type the definition in here. Again, this is a Word document, so you can type right in here. And then we also need to summarize ideas from paragraph 1 and 2 of the procedure section. So this is the procedure section, and there's going to be some paragraphs of information here, and it's asking you to summarize ideas from paragraph 1 and 2. There's 3 boxes here, so you can decide, do I want to summarize 2 ideas from paragraph 1, or do I want to summarize 2 ideas from paragraph 2? But you need 3 total descriptions or summaries of what you remember from those two paragraphs. So what do you think was important from this section of procedures? And remember, the procedures are how the experiment's being designed. How are you setting up the experiment? So just come up with some details that you think would help somebody else remember what's important about the experiment and how it was set up. And that's what you summarize down here. And the only way you can figure out what the procedures are is by going to the page, scrolling down, just like you see here, and you can see right here, paragraph 1, paragraph 2. So read through paragraph 1, decide if you want to have 1 or 2 boxes worth of summary, and then down here, decide if you want 1 or 2 summaries. So what do you think is important from these two? But this is basically explaining how the experiment is set up and how to do it. All right. Once you know that, once you know what the experiment is, and you know how to set it up, the next thing to do is basically start doing what the procedures say. And in this situation, we need to fill up the test tubes with certain species of Paramecium. So, according to this worksheet, it says test tube 1 contains. Let's see if it tells us in the simulation. It says fill the 3 test tubes with, one of the test tubes needs Paramecium aurelia, another test tube needs Paramecium caudatum, and then another test tube needs a combination of both. So we're going to take this literally as this is going to be the first test tube, Paramecium caudatum will be the second test tube, and then the combination of the two will be test tube 3. So to do that, this is interactive. I just pulled in some Paramecium from the flask, and done. Actually, I'm going to reset it because I said Paramecium aurelia will go into test tube 1. So picking it up, so you hit it once to suck the Paramecium up, and then you just let it go. So you click and drag. Paramecium caudatum we said was going to be test tube number 2. So I click it once, and then I click and hold and move it to test tube 2. Bam. Alright, so we're well on our way. And then it says test tube 3 is a combination of the two. So you figure it out. You get one of each, and you put them in the test tube. So test tube 3, so now we know which of the 3 includes. And use your notebook to remind yourself what the dependent variable is, or what you think the dependent variable is. Remember, we say the independent variable causes the dependent variable. And if the independent variable is competition, what do you think the dependent variable might be? And a hint is the type of data you collect. So whatever the data is, or what the data means, that's probably your dependent variable. So take that hint. All right, so once we get the test tubes filled up, the next thing is to, it says third here, it says observe Paramecium and record population data. So right here it just told you the data is the population. And that is in the form of individuals per milliliter. Remember, the Paramecium live in fluid, milliliter, that's fluids. So you pull the Paramecium up, remember they're single celled, they're tiny, they live in the water, so it's individuals per milliliter. It says read paragraph 4 and beyond from procedures to view the Paramecium species population in each test tube. Okay, so I'm going to show you how to inspect the Paramecium. Alright, so you click right here. So you click on the microscope, and you're going to, let's see here, it highlights, so it lets you know which things you can interact with. So the next thing is you click the microscope slides, and this guy right here is going to get, let's see here, oh, here we go. Notice it only filled 2 of the 3 microscope slides, and that's because I haven't technically put anything in this third one. Remember, this third one's supposed to have a combination of the 2 species. But this is just for examples. I'm just showing you the process of how you actually view the Paramecium. So this is day 0. So let's look at the data table here that you're going to be filling out. Worksheet. Here we go. So the data table looks just like this. So your test tube 1, remember, has P. aurelia. Test tube 2 has P. caudatum. And then you have both species mixed, right? So let's look at day 0, and we're going to figure out how to fill this thing out. So we clicked on the microscope, and then we clicked on the microscope slides, and then just by clicking on the test tubes, it will automatically fill each slide based on your test tube. Now we didn't put anything in test tube 3, so this is blank. Test tube 1 has P. aurelia, and you take it, you click and hold it, and you leave it with the microscope. And you can see right here, on day 0, you have 1 P. aurelia. And so in your table, you would put uno. Let's see what we get for the next one. And you just click it, and it'll swap out automatically. And you have P. caudatum. You can see their shape's a little different. P. aurelia looks like kind of a sesame seed, and P. caudatum is a little more stretched out, a little longer, okay? And you can see that there's 1 P. caudatum there. So we have a beginning population of 1 of each. There you go. Simple enough. You just type in the numbers as you see them. Now, if we had test tube 3, which is both species mixed, you'd have a number for each of these. I'm guessing it's probably starting with 1 for P. aurelia and 1 for P. caudatum. Now, the reason why the population can increase from 1 is because they basically do cell division. They don't have to mate with another individual to reproduce. They can just split themselves, or because they're a single cell, they can do cell division to reproduce. So it's kind of asexual. All right, so, and then you would go on to day 2. So I just want you to see what day 2 would look like. So let's see if we can figure out how to do this. Whoop, that's not it. So let's see if it, oh, you got to clear slides. So once you hit this button right here, clear slides, you then can go on to day 2. All right, and then we repeat the process. You click on day 2. So you see I hit clear slides, and then I went to this thing that highlighted. It tells you what you can hit, right? Now it's on day 2. I get the microscope slides. I go to the sample, the test tubes. And now I can go, and I have P. aurelium here. I drag over, and now there's 4 of them, right? All right, so then you would put, for P. aurelium, you got 4, right? Now, the next part I'm going to show you is this hint right here. There's 4 now, and it's easy to count them. But by the time you get to day 10, 12, 14, or day 16, which means you repeat this basically 8 times total to get to day 16. It skips a day, so it's even days, day 2, day 4, day 6, and so on. By the time you get to the higher dates, day 10, 12, 14, and 16, it can get hard to count these. And yes, you're expected to count them all. So what you do is you click on the grid. And the grid basically helps you kind of stay organized as you count them. And you just continue with the process till you get through with day 16. Let's hurry up though, and let's move and look at a little bit more of the worksheet. So you fill this whole data table in, and then it tells you how the populations did when they were alone and how the populations of each species did when they're mixed. So this is your raw data. This is the information you can use to understand if your hypothesis was correct, your initial one, but also to answer questions below. And that's the part where the analysis part of the experiment, where you try to make sense of your results. So let's look at that. Okay. Method says how experimental design provides results and knowledge. So what do you think the control group was? Which test tube do you think that was? Or is there multiple controls? The experimental group contains the independent variable. What do you think the independent variable was? And then how did you use the control group and the experimental group to answer the first question from page 1? And that's the hypothesis. So how are you answering the hypothesis and the question using these ideas of independent variable, dependent, control, and experimental? So this question right here relates to the very first question or issue, which was the very front of the page, which was how does competition affect population growth? So use those ideas from chapter 1, and that's independent, dependent variable, control, and experimental group, to answer that question. So use the ideas of experimentation to answer that. And then you're really at this point just reporting on the results. When do you think Paramecium caudatum and Paramecium aurelia reach their carrying capacity? And at this point you should know what that is, but if you don't, you can Google it or look it up in the textbook for chapter 4. You'll compare, contrast some of your results. And then the last section here, and I'll just say this briefly, is you need to summarize and use information from question 3 here, as well as this last section, information here, to try to make sense of your results. And good luck. All right, try your best.